Good morning, everyone. Thank you for joining me today as I present my research on how the invasion of brown seaweed 𝘎𝑰𝑚 affects the restoration efforts of the endangered Gongolaria nadicalis. So for this, we're gonna follow this plan, introduction and general context about biological invasion, restoration process, and we're gonna discuss our results, and then the presentation with conclusion and some perspective. So, the biological invasion involves plants, animals, and microbes. The process by which the native species are introduced, become established, and spread and colonize new region. Invasion overcame natural barriers and it can outcompete native species for space and resources. So the impact of biological invasion in general, it's about losing biodiversity and declining of native species, also affecting global marine freshwater alternate ecosystems function like nutrient cycling. The major impact of this biological invasion it's about its own restoration challenge and natural recovery. So the brown invader species in Morocco and for our study is Sargassum mythicum. It's an invasive brown seaweed native to the Pacific Ocean. It's also reported in Morocco in early 20s and it was introduced in Morocco via oyster farming and also spreading via hole falling and ballast water and ocean current eating its dispersal along the Moroccan Atlantic coast. Many factors aid the adaptation of the invasive species, known for its rapidly colonizing coastal area, posing a serious threat to native marine species. The brown native species in Morocco are many. One of them is the Cistos genus. This genus is a group well known for its significant role as a key forest forming alga. So alga base lists around 16 species in Atlantic and Mediterranean Ocean, with just 4 species are mainly Atlantic. The the native fucoid studied or the impacted species for our study is Gongolaria nadicalis. It's a brown seaweed and native to the Atlantic Ocean. First occurrence was noted by our team in 𝘎𝘤𝘪𝙩𝘜 coast, and also its ecologically significant species, serving as a niche for a diverse epiphymal biodiversity due to its complex and spiny structure. So, given the role of Gongolaria nadicalis as also a bioindicator of water quality, the species has recently declined significantly by the aggressive spread and competitive dominance of Sargassum mythicum, which poses a major threat to its survival. As a remedial measure, our team has initiated the first attempt to restore the species. In parallel, we aim to assess the impact of Sargassum mythicum on the recruitment and developmental dynamics of Gongolaria nadicalis juvenis. So for our restoration process, this slide outlines the 4 key steps for our restoration process into the impacted site. The step 1, it was about harvesting the fertile branches from the donor population, followed by careful transport to the lab. In step 2, we maintained the zygote from these branches in the controlled condition in the laboratory for 26 days, allowing the development of juveniles individual on tiles. The step 3 involved the outplanting of the style at the recipient site via their screwing on bedrocks. 2 experimental conditions are tested: one with the presence of the invasive species Sargassum mythicum to simulate occurrence, and one with its absence to evaluate the potential for successful recruitment and growth without competitive interference. The final step is monitoring this outplanted tile over a period of 3 months to assess survival and growth performance under these 2 conditions. For the results, so like you see, this graph shows the survival or the density of juveniles over 5 time points in 2 experimental conditions. By 0 time, both the cleaned zone and the zone with Sargassum mythicum show high juvenile survival. After just 15 days, the density drops sharply, especially in the impacted site. Over time, the control site or the cleaned site maintain better survivorship, while the impacted site continues to decline more steeply. By the end of 3 months, only about 10% of recruits survive in the impacted site, but 14% in the cleaned zone are survived. This clearly shows that the presence of 𝘚𝘢𝑚 mitigum strongly reduces recruit survival, and of this may be due to to space competition, and also to release of allelopathic substance that negatively affect native species establishment and growth. So for the length or the growth of Concholoria nadicalis juvenalis in these two conditions, at zero time, both groups started with similar length, but by just 15 days, juveniles in absence of 𝘚𝘢𝑚 mitigum already show better growth, almost double that one occurred of the impacted group. This early gap suggests competition likely for light or space. So by 1 month, the difference became more obvious. The control group reached nearly about 17 millimeter, while the 𝘚𝘢𝑚 groups remained below 5 millimeter, suggesting possible chemical inhibition from 𝘚𝘢𝑚 on Gangularia nadicalis juvenalis. By 2 months, the control group kept growing while the impacted group barely advanced. Finally, at 3 months, the cleaned zone or the cleaned zone reached about 26.7 millimeter, while the impacted zone it was stuck at 7 millimeter, less than third. This result clearly showed that 𝘚𝘢𝑚 severely reduces juvenile growth, likely through shading, space competition, and allelopathic effect. Managing these invasive species is essential to protect native algae and biodiversity. And to conclude, positive survival and growth results suggest that ex situ cultivation is workable for restoring Gongolari nadicalis population. 𝘚𝘢𝑚 mitigum disrupts recruitment, resulting in decreased density and size of affected population, and further research is needed to test on different type of substrate and in another area. So thank you all for your attention and interest in this work.